So Ertugrul is not a visitor from far away. He's on the faculty in Feinberg, so he came from his office downtown, I guess. He's in the Department of Cell and Developmental Biology in Feinberg, in the Feinberg School. But he's a physicist by training, undergrad and grad school. He did his PhD at MIT with Alexander, what is it? Udin-Norton. I'm gonna ask for his name. But so he's worked on stochastic gene expression. He's worked on biological oscillators, which is something I share an interest with him in. He's also a Sloan Fellow. So I'm looking forward to hearing more about his research here. Oh, and I should say he is in Feinberg, but he just joined Feinberg in the summer. So he's still relatively new to Northwestern. So welcome, Ertugrul, and thank you for your talk.
Thanks a lot for the invitation. So in my lab, we are interested in developmental pattern formation. So if you zoom into developing embryos, you'll see that groups of cells break symmetry and establish characteristic unique patterns that is specific for each developing tissue and all. And we are studying this broad question in the context of segmentation of the somites. And these cells in these somite segments, they are the baby name somites. They will later on give rise to the vertebral discs. That's why we have these vertebral discs segmented from each other, instead of having an unbroken concrete pillar. Okay. It's important for the motility for all vertebrates, hundreds of thousands of species. So the number of vertebrae varies more than tenfold across all vertebrates, but in a given species, that number is quite precisely fixed. So it's a very precise, robust developmental pattern formation. And these vertebral discs are forming sequentially in vertebrates. What happens in all vertebrates is that the body axis from head to tail elongates initially in a unidirectional manner from the region will be corresponding to the future tail or fin. So we use zebrafish as the model system in our lab. So this is a zebrafish embryo, bright field pictures. As you see, the axis elongates downwards posteriorly from their tail end. As the axis elongates, it forms somites sequentially. Those chevron-shaped borders in the bright field pictures are separating one somite from the other. And this process has an approximate periodicity, which is species specific, happens every 30 minutes in fish, up to 5 hours in human embryos. So if there's a periodicity in pattern formation, you would expect to see a biological clock, and indeed that's correct. So this is the same animal now, but we are also now, I'm releasing to you the transgenic reporter on the left, which basically shows you one of those segmentation clock genes, pulsatile, expressed in this unsegmented tissue. And the tempo of the period of this clock oscillations is the same as special pattern formation. So this cartoon, sorry, I went too fast. So this cartoon summarizes what happens in this movie as well as in all vertebrate species. So the cells located in this tail end of the embryos, in the trisomitic mesoderm, they express a small set of genes in an oscillatory manner. These oscillations displays moving kinematic waves from the tail end posterior towards anterior end of this tissue. After one clock cycle, this basically picture will rewire back to the first initial snapshot, with the exception that at the anterior end of this unsegmented tissue, a pair of somite will be segmenting, one on the left, one on the right side of the spinal column, which is in the middle.
And that's one cell only on each side?
No, these are one somite. Each somite has hundreds of cells in it. So these waves are kinematic waves, as I told you. And if you look into which genes display oscillatory expression in this unsegmented tissue across all studied model animals, vertebrate animals, we'll find that the oscillatory dynamics of 2 families of genes are conserved, only 2. One of them belongs to her family transcriptional repressors. I'll talk about them in more detail later. The other group belongs to delta family ligands, signaling pathway ligands. And mutating these genes, these 2 gene families, or preventing their oscillatory expression by driving them continuously express, in both cases, it results in segmentation defects, okay? The vertebral column will be not segmented properly in all studied animal models. It also results in a birth defect in human patients, congenital sclerosis. So this is a human patient picture. So this is the head, these are the ears. This is supposed to be the vertebral column. Instead of having nicely organized discs, this has random incomplete fractures and kinks in the 3D, okay, which has a mutation in one of those gene families in this patient. So what is the pacemaker mechanism of this clock? In this second introduction slide, I'm going to introduce you that. So if we dissociate cells in this unsegmented tissue and culture them as single cells with fluorescent reporters, you'll see that single cells display oscillations, which means the pacemaker mechanism must be cell-autonomous. Several conditions theoretically needs to be satisfied to generate a limit cycle and an oscillator. And we have experimentally tested those conditions and validated it. In zebrafish, the first one is you need a negative feedback loop. In zebrafish, two genes, HER1 and HER7, they act redundantly, additively, and they belong next to each other on the same chromosome, divergently transcribing gene pairs. What we have found is that they produce proteins. These proteins act as dimers. They dimerize with each other. And those proteins auto-repress their own transcription, like a snake biting its own tail. They have a feedback loop, satisfying this first condition, this first requirement. But for these products of these genes, RNA and protein, to be oscillating, they need to be extremely short-lived so that they can oscillate every 30 minutes. Again, from theoretical studies, we know that number should be very small, less than 6 minutes. We experimentally calculated, measured their half-lives, and we found them to be on the order of 3 to 5 minutes. The third necessary condition, the feedback should not be instantaneous. There should be sufficient time delays in the system, and there should be adequate non-linearity in the system. The non-linearity comes from the dimerization of these proteins. And we also calculated the transcriptional time delay to be on the order of 7 to 8 minutes. So if you now write mathematical equations describing this auto-inhibitory negative feedback loop with proteins forming dimers, etc., and then simulate it with these calculated parameters, then obviously we don't know every biochemical parameter, but this is a small network, not too many other free parameters. Then you can recapitulate, find several parameter sets that successfully recapitulate 30 minutes rapid oscillations in silico that we observe in vivo in the intact embryos. So you ask a nice question, somite do not form from single cells, groups of cells forming a somite, so there should be a cord or team effect. You can also see at the tissue level, if you look into this tissue, you'll see groups of cells in the on phase, let's say highest expression, orange. Others are in the light pink, lowest expression, freezes. So you'll see local synchrony among neighbors in the short spatial distances. And that's important because these groups of cells will be committed to as a team into a somite. So how is that local synchronization is accomplished, right? You're burning to know that.
I am.
I was just going to say, but your question before that.
I have a very simple question.
Mhm.
So what's the connection between the left and right symmetry here? Are those pieces connected in outside of the plane of the projection?
They are not. So those cells were connected when they were in the tail tip. Once they merge some of them stochastically to the left or to the right pillars of the tissue, they no longer communicate with each other. There are other tissues in the middle, as I told you. Neural tube is in the middle, which will later on make the spine, and notochord as well. They separate those tissues from each other. They were once upon a time together at this tail tip. But until the exit of the tissue in wild-type embryos, they maintain left-right synchrony. They don't deviate enough. In other words, there's not that enough noise to disturb them from each other or separate them from each other. And segmentation is, for the most part, also happens left-right symmetrically. Yeah, there could be minute seconds and stuff differences, but yeah. So how is that local synchronization is accomplished? So you need cell-to-cell communication, and that's accomplished by delta notch signaling pathway. And that's among the signaling pathways is unique because the ligands of this pathway, delta, is membrane bound as well as the receptor. So it's not a secreted molecule. It only operates in short distances. Cells touching each other or expanding philopodia, then they can touch maybe a second or third diameter of the cell's maximum. Okay. Again, we have experimentally characterized this system, also did computational simulations on it. Again, if you add this now, delta notch coupling, and simulate them in silico, you can see cells are synchronized together when there is this communication between them. What's the role of the delta notch? If we mutate these pathways genes, it results in birth defects in patients, as I told you. If you then image intact embryos containing mutations of these genes, you'll see that with the fluorescent report, single cells still oscillate. Now they are no longer synchronous. And it gives you like a salt and pepper pattern in individual time snapshots at the tissue level. Okay. So it minimizes cell-to-cell variability, prevent the drift of oscillation phases from each other, and maintains them in synchrony in the short distances. We still have ongoing projects on this genes, cell-autonomous proteins, clock genes, as well as the synchronization processes. I'll share two completed story in the coming slides to you later. All right, so we've been investigating this regulatory network for over a decade now, and then published several papers, still projects ongoing. I'm going to share one of them to you quickly now. We were investigating role of positive feedback, negative feedbacks in this system, dimerization, etc. And we stumbled upon one feature, which is called divergent gene pairs. Okay, if you remember, these two genes are one another next to each other on the same chromosome, transcribing opposite durations. So that term, divergent gene pairs, originally is described by a bioinformatician studying initially his genome, which is a very compact, small organism, small genome. And they arbitrarily, subjectively describe those genes as transcription start sites separated 1 kb from each other. There is no objective experimental criteria for that. With that criteria, they find 50% of the yeast genes belong into this category. Again, with that very restricted definition, 30% of fly genes, 10% of human genes fell into that category. Obviously, in higher metazones, genome is large. You have to relax that. Then probably the percentages will be similar. The second thing they have done is that when they compare these genes expressions, they find that these divergent gene pairs in the transcriptome level statistically significantly more co-expressed as compared to two randomly picked genes on the gene. They also published data on that, again, statistical analysis, bioinformatics. But again, we do know that up until now, we didn't know really the functional benefit of maintaining such genes. The reason is that, so first of all, they also characterize which gene families they belong. Many of them belong to housekeeping proteins, important for the essential of the cell life. But some of them also code for specific roles, such as transcription factors and signaling regulators. And these capitalized names are coming from the human genome, whose transcription starts are separated 1 to 500 kb from each other. Okay. So now, again, from transcriptome studies, we do know that you do not need two genes next to each other for them to be co-expressed. In fact, hundreds of genes distantly located from each other on the same chromosome or even on different chromosomes, they can be pulled together in the transcription hubs or condensates. There are several other regulatory mechanisms ensuring their co-expression. The only thing they need is that they need some common upstream regulators, right? Then you can drive hundreds of genes, co-expression of hundreds of genes. If that's the case, which is the case, then why some genes are still maintained next to each other? Is there any additional advantage of maintaining gene pairs or divergent gene pairs on the chromosome? This was not experimentally being tested, and we wanted to test it by using two segmentation clock genes in zebrafish as the test bed for this question, whose transcription start sites were separated 12 kb in zebrafish. Okay. So to be able to test it, we did several experiments. The first one provides a bit correlative data, single molecular fish experiments. If you are familiar with it, you basically label individual RNA with very bright fluorescent probes. Then it will allow you to see nascent transcription loss and active transcription on the genome. And when we do that and we look for these two genes, two chromosome copies, so 4 loci, what we find is that two different genes on the same chromosome co-fire slightly more frequently as compared to two same genes on different chromosomes. Again, two same genes have the same promoter, same regulatory elements. They're just on different chromosomes. They're also co-firing because all 4 copies are responsibly repressed by the same repressor. But two different genes on the same chromosome still have as an edge the advantage of being co-fired slightly more. And it's a bit correlative data. So wild-type embryos successfully segment their somites, so we can't really assess it. We had to sensitize the system. So to be able to do it, we basically generated double heterozygous mutants. So we mutated one gene copy, and we basically generated 2 different variants of this by using CRISPR-Cas9-based genome engineering. So in the first case, the stars mark the mutations. We mutated one HER1, one HER7 in the same chromosome. The remaining functional genes are still in the same chromosome. They are still paired. We call them gene-paired embryos. In the second case, we again mutated one HER1, one HER7, but this time the mutants are on different chromosome. The remaining functional genes are on different chromosome. They are gene-unpaired embryos. Okay? These are loss of function mutations early in the coding sequence. They are null mutations. We know it because if you mutate all 4 copies, it's equal to chromosomal deletion of all 4 copies. So it's, function of the protein is lost when they are marked with this star, but only on that copy, not the unmarked gene copies. Okay. If you pay attention, we are not changing the number of or identity of the functional genes. In both cases, we have one functional HER1, one functional HER7. We do not disturb negative feedback. We do not disturb protein structure, RNA or protein half-life. Nothing changed. The only difference between them is being on the same chromosome. So now if you raise them and quantify segmentation success by marking segment borders in individual embryos with a marker. So now this is what we got. In most of the gene-paired embryos, almost all the segment borders successfully form without any disruption. The opposite is true. Overwhelming majority of unpaired embryos, they have randomly distributed disrupted segment borders. In this particular embryo, in the middle of the axis, it has broken fragmented borders. Again, we can quantify this significantly different. Overwhelming majority of these guys successfully segment all boundaries. Did I touch something? I don't think so.
Computer doing.
Yeah.
Is it on the computer?
It's on the computer screen.
Maybe it's on the computer screen.
Maybe your Zoom crashed or something.
The Zoom crashed.
Oh,
Yes.
Naturally.
Is there a Zoom listeners?
Well.
Restart.
Good. Let's play that.
Are we back?
He's in Zoom, yeah.
I am in Zoom. I am sharing
Okay.
Zoom. I don't know whether Zoom is up here. Should be, otherwise I wouldn't have joined it. Right?
Yeah, then I should be good. I'll let you know.
Okay. Now this is not working.
Yes, please.
We just love to torture the speaker.
no.
It's okay.
It's like,
You just get it away. Okay, now it's working.
Wait, we lost the shared screen.
it's back, yep.
Okay.
All right. So, all right. You got the summary. Okay. So this is the very first experimental evidence of gene pairing providing an extra edge of advantage of co-expression of genes and being important for pattern formation. But again, we ask where is this advantage coming? Obviously, we suspect co-expression of genes, but so in the wild-type embryos, obviously you have a limit cycle, RNA and protein levels oscillate in a circular manner. If you plot them in time, you have oscillations. If you plot them in tissue level in space, you have these moving kinematic waves. In the homozygous mutants, where all gene copies are mutated, what happens is that you are locked in a stable state. The levels are intermediate, moderate level, it's locked. In space, kinematic waves are lost. You have non-oscillating uniform expression in the tissue level. But our mutants are neither of them. They are heterozygous mutants. There's still two functional gene copies, right? Why the gene paired embryos most of the time behaves like this, up row, and the gene unpaired embryos occasionally, most of the time behaves like this, right? That's the question we are trying to address. Again, from modeling, we know several conditions needs to be satisfied to generate an oscillator, but we didn't violate any of those. But I hit the last condition though, so there's one additional condition needs to be satisfied. The production rate needs to be very strong to overshoot the intermediate steady state and establish a limit cycle. So because from our SMFISH data, we do know that when the two genes are on the same chromosome, they are more synchronously transcribing. What we think is happening is that we are violating this condition in the when the genes are unpaired. In the unpaired embryos, occasionally one gene is turned on, the other one is not yet. We are not accumulating adequate amount of total RNA, and then not enough total protein. And occasionally in some cycles, you lock into this intermediate state until you recover back later on. Whereas in the gene paired embryos, because you are ramping up transcription of two genes simultaneously, then you overshoot this intermediate state and establish a limit cycle. Very rarely you fail to do that, but it also fails occasionally. Okay, so we did some stochastic simulations that agrees with our predictions, where we give the propensities here that two genes in the paired cases fire together as compared to a random number. In this case, we have two different random numbers for each separate genes because they're on different chromosomes. And the correlation scores of these two genes decreases when they're in the unpaired configuration, as expected. Again, SMFISH experiments agree with it, with mass and loci being both transcribing in the gene paired configuration versus unpaired configuration. But we wanted to further test it by doing live imaging. So we did now segmentation called protein level imaging. We also took bright field pictures so that we can score segment borders. So disrupted borders we mark with star. So we imaged three different genotypes: wild-type embryos, gene-paired embryos, gene-unpaired embryos, starting from 15 somite and ending at 25 somite, 11 somite cycle. So these are the amplitude of oscillations in wild-type embryos. You have 4 functional copies. Obviously, the amplitudes are always good. In the gene-paired embryos, many boundaries cycles amplitudes are good, occasionally disrupted. The opposite is true. Many are disrupted, occasionally they are good. in the gene unpaired cases. If you quantify the cumulative amplitudes, the wild type, obviously with 4 gene copies, all amplitudes are high, but apple-to-apple comparison can only be made with this green and orange population. In the gene pair configuration, the cumulative average amplitude of oscillations across all cycles, across all embryos are higher compared to gene unpaired embryos. And their segmentation success is also very close to wild type, higher than gene unpaired embryos. Now, if we split these amplitudes into 2 groups in each genotype, because we have time-lapse movie, right? We can say, what's the amplitude of oscillations preceding failed segment borders or preceding successful segment borders, right? In both cases, what we find is that the amplitudes preceding normal segmentation are higher than the one preceding disrupted segmentation. But that successful higher amplitude of oscillation happens more frequently in the gene pair configuration. That's why they have higher number of cycles they succeed generating somite segments. Okay, so this was the very first time showing that gene pairing advantages for biological function, particularly developmental pattern formation. But a group after our paper is published from Princeton, Mike Levin's group, I'm not sure if you know him, studying Drosophila segmentation genes, they also cited to our paper. They also found that when genes are together on the same chromosome, their co-firing of transcription is higher. So we have at least now n equal 2 examples. All right, so up until now, I have spoken to you about the clock, the tempo of pattern formation. But the embryos also need to know how many somites they will form, how many rows of cells they will allocate to each somite, right, in a fixed body length. So in other words, they need to know position information, where to place segment borders. Segment border information needs to be discretized in the space, right? Several rows will be passed over, a passover phenomenon, and then a border will be instructed across this row of, or this disk of cells in 3D. So that position information is provided by long-distance acting signaling gradients. Fibroblast growth factor and its major readout is double-phosphorylated ERK kinase. They both establish a gradient posteriorly high, anteriorly declining, mimic with this blue color in this tissue. And several groups have shown that if you manipulate this gradient, if you suppress it towards more tail or expand it towards more head, you can change somite sizes. Okay, so we do know this gradient provides position information. Again, double-phosphorylated ERK kinase is the readout of this pathway. It's the one instructing the segment borders. Okay, so in our field, over the last half a century, we have a, go ahead.
So if you have longer regions, bigger somites, do you kind of grow longer, or you just feed fewer because there is something that controls how long you grow? So
if you can make it continuously longer, you don't grow longer. You just will make less.
Okay.
Yes, good question. So one quick question about this gradient establishment is, in case you wonder, so the tail of the embryos keep elongating, as I told you, right, in all vertebrates. The elongation happens because new cells are diving into the tail tip here, this tail region area, and they hijack the tip, okay? The cells that are previously dived into the tissue, they are kicked out, and they are basically joining to the left and right columns of the tissue. So there's a space-time ordering in this tissue. The more closer you are to the head or last segmented somite, the earlier you join into this tissue, the longer closer you are to the tail tip, the most recently you join into the tissue, okay? And only cells that are spending in this tissue tail tip for a short duration, only those cells are actively transcribing the ligand RNA. Once they are kicked to the left or right column, they turn off, switch off transcription. That's why there's an RNA gradient that's established for signaling, which converts to protein gradient, which then converts to the readout double-phosphorylated ERK activity kinase gradient. Okay. So in our field, how this temporal and position information are integrated, up until last year, we published a paper, it was unclear. We had an abstract phenomenological model lingering in our field, dominating over half a century. And it had 3 untested assumptions. It was stating that this gradient is static in the moving, elongating tail bud reference frame, like a passenger sitting on a seat in a moving car. Okay, the peak level of the gradient doesn't change, the spatial range doesn't change, the slope of the gradient is not changing. It's just passively moving as the tail elongates, it drags the gradient away. And it provides position information at an unknown arbitrary concentration. In this cartoon, it's subjectively placed here, this horizontal dashed line. Okay? Whichever cells smell that concentration become compatible to make a segment border. So you can easily see that because tail elongation is continuous and the gradient is continuously regressing, this front, which is called wave front, is also continuously regressing like a wave over the cells, it's swiping over. So why don't we have then somites forming single cell diameter? Right? How do we get this discretization or this passover phenomenon? Several slices are passed, skipped, then a boundary, a slice of cells is extracted for a segment boundary. So to be able to explain that challenge, the model also speculate that, well, temporal and spatial information are provided independently, and the cells somehow integrated like an AND gate in engineering. Some common targets, perhaps, receiving input from both. So that's why this gradient gives a permissive cube, but as it passes, none of those cells are instructed until they also receive a second coming cube later, the clock array. Only the cells that are sensing an unknown phase of the clock, at the same time simultaneously an unknown concentration threshold of the gradient, are to be chosen as the lucky cells to make segment borders. Okay. Again, it's an abstract model. It's very difficult to even simulate it. You can't simulate because it's not molecularly making any predictions. Right? It's good to be vague and abstract. You can't stay half a century long in the field. And it had influenced us a lot. So, we are grateful for those great giant scientists who had proposed this idea half a century ago, but it's no longer suiting anymore to this body. We need a new clothing, basically. So over the last 6 years, we tested these 3 untested assumptions, and we find all of them to be wrong. I'm not going to show the first story we published 6 years ago. I'm going to show the more recent one that we violated 2 of it. And then with that, we are basically confronted with the challenge of proposing a new model. Right? If you basically kill the model of your scientific grandparents, now it's your responsibility to fix the mess that you created. Okay. So to be able to test those 2 assumptions, we basically, for the first time, imaged simultaneously segmentation clock as well as the ERK kinase activity. So they had been quantified independently, direct or indirect readouts, but not together. So I'm going to start the movie and then pause and then explain the reporters. And then, okay. So we did 4-channel imaging. So the top 2 are membrane on the left, nuclei on the right. The middle is segmentation clock, which is a transcription factor. It's mostly nuclear localized, as you can see, but it makes kinematic waves. You don't see every cell is bright. On the middle right, we have the ERK kinase reporter. ERK is a kinase. Its phosphoform is active. Making a reporter for a phosphorylated protein is not easy. You can just get gene under a GFP or promoter or something. Fortunately, the technology was developed at Stanford 10 years or so ago, called kinase translocation reporter. So ERK is a phosphorylated ERK is an active kinase, phosphorylated hundreds of proteins. So they took one of the target peptides known, a small portion of it, which has ERK docking on phosphorylation site. Engineer nuclear import-export signals in it. Afterwards, you can find any fluorescent protein. In this case, we use PACDA. Now, in whichever cell there's a high phosphorylated ERK protein, this synthetic reporter is phosphorylated by ERK as among its targets. It exposes nuclear export signal and keep this reporter out from the nucleus. So posterior is the right. So because ERK gradient is high in the posterior, you see black nuclei mostly in the posterior. In the middle, you see more uniform signaling because as ERK decreases, some of the reporters are allowed to go back into the nucleus. Obviously, to be able to quantify ERK activity, one has to calculate cytoplasmic to nuclear ratio of this reporter in single cells in a compact tissue. That's why we need a membrane and nuclear reporter and a lot of time to optimize imaging and image analysis. And now, once you do it, so this is the ERK activity post-analysis. All right, so you can follow a population of cells like this yellow circle at the tail end. And we see that clock activity oscillates as expected. But we also find, to our surprise, ERK activity oscillates as well in zebrafish, okay? So these are the single cell tracks instead of that cell population. Clock segmentation clock already known to oscillate for quite a long time. But we also find ERK activity also oscillates with the same period. Their oscillations separated by pi. So they're oscillating anti-phase. The reason they are oscillating anti-phase, we have a paper just recently accepted in developmental cell. Hopefully it will be coming out after Thanksgiving online. What we find is that segmentation clock proteins inhibit ERK activity. So the clock auto-inhibits itself, drives its oscillations. But when the protein levels are high, it also inhibits ERK, decreases ERK. When it self-represses cell goes down, ERK recovers back. And that's why they're oscillating in an anti-phase manner. So this violates the first condition. The gradient is no longer static. In every embryo, there's a special gradient like this, high in the tail end, lower towards the head. But the slope of this gradient oscillates. It never becomes flat. It goes roughly down to 50% or so. Okay. due to periodic inhibition by the segmentation clock. How do we know that? Because of the upcoming paper, but also in the segmentation clock mutants where 4 gene copies across 2 chromosomes are mutated, all oscillations are gone, all segment borders are disrupted. In that background, now the ERK activity gradient stops, oscillation stops. It becomes a static gradient in space like in the textbook model. So it also violates the second assumption. The dynamics of the ERK activity depends on the functional segmentation clock. They are no longer independent, and this is no longer a static gradient. There's a gradient in space, but there is a temporal oscillation in the amplitude of this gradient or the peak level of this gradient. And the slope of the gradient also oscillates. Okay. All right, so these were the violations of the new model. And we also find for the first time a molecular hierarchical connection between them, which is amenable for computational modeling and experimental further testing. So now we need to propose a new model, right? So we have challenges to overcome. One of those challenges that I will emphasize is in cell biology, we do know that several pathways crosstalk with each other. So there's lots of learnings going on, but not always those crosstalks will result in stable functional outcomes. Okay, sometimes they are useful. There's a reason why one pathway crosstalk to the other. Many times they are irrelevant. The cells just tolerate it, or they filter it out by using several genetic circuitries. So how do we know that segmentation clock induced ERK activity oscillations in the peak level is functionally important? So our model needs to be able to accommodate. We also didn't want to propose yet another abstract phenomenological, untestable, unsimulatable model in the system. We wanted a mechanistic model. We can simulate and test it. So to be able to do it, basically, we get inspired from one of our favorite physicists, Richard Feynman. He had this famous quote that became the motto of all synthetic and systems biologists, biomedical engineering, bioengineering, chemical engineering folks across the globe. So inspired from this motto or quote, if we convert or reflect that statement into our system, so we realize that until we take a genetic mutant on its own incapable of segmenting, then combine the temporal and spatial information in a manner predicted by one unique model, and re-engineer missing segment borders. Until we accomplish that goal, we will fail to reach a mechanistic understanding of this beautiful, famous landmark example of developmental pattern formation. So that was our goal that we tried to accomplish. With that, we proposed this new model called clock and oscillatory gradient, in short, COG, like the cog wheels in mechanical clocks. Hopefully it will stick in the minds of the people in the field. According to this model, segmentation clocks' only role is to induce ERK activity oscillations. Remember, it's a transcription factor. It has hundreds of targets. What we claim now is that it has basically 2 roles. One is to self-repress to drive its oscillation. The second one is inhibit ERK activity. So long as it does that, the rest of the targets are irrelevant. A very bold statement. And if this is true, then we should be able to test it. Okay? I'm going to give you the animation of our model here first, and then I'll tell you how we test it. I'm going to also introduce you some biology again further here. So the tail is, as you see, left. It's growing posterior leftwards. These rectangular blocks are segmented somites. I'm only showing the last 2 of it in the beginning of the animation. Moving kinematic waves of the clock are yellow. The gradient is red at the top. Wherever the clock waves moves, it inhibits our gradient and decreases it. And there is one important thing that I need to maybe introduce you. And this is coming in all vertebrates, not just specific to zebrafish. The clock and the gradient provides information in the middle of the tissue, where this black star is marked, and also a vertical red dashed line. This is the place where the action is going on. Anterior to this location, these cells with bright field pictures, you cannot see the fissures like in the segmented somites. But these compartments are already time-stamped, predetermined to segment. If you disturb the clock in a time-controlled manner or disturb the gradient, you cannot stop them from segmentation. You cannot change their sizes. They become independent of the clock and the gradient and the cog model. They just execute segmental decision due to biophysics of epithelialization and segmentation process. The temporal and spatial information are already delivered to them. They're already separated from each other in their mind, although we cannot see it, okay? And that actually is happening here, yes.
So the key thing here is that the period of the clock matches the period of the segmentation.
Yes. Approximately, yes.
Okay,
Mhm.
so I mean, both of those have presumably been measured before
Yes.
and they were known to be
Like,
similar?
approximately, yes.
Okay.
Mhm. So that's our model. So again, if our model is correct, and if this dynamics of ERK is really important, and this is how temporal and spatial information are integrated, and how the information is discretized and this position, once a wave passes here, clock wave, the color flips from red to white, and now the next position will be more posterior. If these are all true, we should be able to experimentally test it, but after this question.
What's the characteristic diffusion time for like ppErk?
The ppErk is a still autonomous molecule. It's a readout of the signal. It doesn't diffuse. The ligands are upstream of that
Okay.
kinase pathway, but ppErk does not diffuse.
What about the things that produce it? I mean,
The ones that are diffusing? Our estimate, so it's not measured experimentally. Based on some experimental data and simulations, our predictions was, if I remember correctly, 50 micrometer per second. That's what I remember, if I remember correctly.
Excuse me?
Yes.
Okay, so if I want to have a certain size of each segment, I guess I can control both the period and also the speed.
Yeah.
I'm curious, what's
Several
the
things you can
speed?
control. One is the clock period, one is the gradient activity, if you can shift it, or tail elongation. Indirectly, you affect the gradient. So there's several knobs you can theoretically manipulate it. Manipulating things experimentally had proved to be difficult. We have some mutants where clock period slowed slightly, although not immediately directly measured in all cases, but at least predicted to be. And we do now see larger somites. And on the whole body, they generate one or two less somites. Okay. The other way around, experimentally, we can manipulate the gradient, as I told you, but in a short time window. And then we can see, immediate effects. But then because we can't do it longer for other restrictions, then the system recovers back after that. But locally, we can, there was one question and then come back to you.
It's very interesting talk so far. So I'm missing something about the cog model. So how is the double-phosphorylated ERK determining when you get a segmentation?
You mean border?
Yeah.
I was going to hide that question. I was hoping not ask, but you ask. Shall I address it?
If you're going to get to it later, I'm fine
Yeah.
waiting.
I'll
I
give
just
you
wanna
a
make sure
teaser
what's-
now, and then if I have time left, I'll show you additional appendix slides.
Okay.
So I didn't mention, as I told you, there was 3 untested assumptions. I show you data disvalidating 2 of it. The third one I said I'm going to skip. It was 6 years ago published. It disvalidated the assumption that ERK was instructing at a fixed concentration threshold. What we find is that ERK is instructing segment borders with a fixed ratio of ERK activity between anterior posterior slices of cells. So that's why these oscillations are important, because both the slope of the gradient oscillates as well as the row levels, and they cancel each other. And that ratio, despite this dynamic change of ERK activity, dropping from this original gray position to the red position, the red color stands still here. The star mark stands still here because the ratio stands still there until another clock wave comes, wipes it off, and it jumps to a more posterior location. Yeah, because position information is encoded not at absolute levels. Otherwise, it wouldn't be kept constant in that location. Neither in a static gradient because tail elongates, nor in an oscillatory gradient because now on top of it you have an oscillation. But because the information is provided in a ratiometric manner, and there is a special gradient, and they're both inhibited proportionally by the clock wave, now you maintain position information in one location, and then it jumps in the next clock cycle to a more posterior location. Okay, I can show more data or slides if you want it later in the Q&A if we have time left session. But we have one question here.
Yeah, just kind of following up on Daniel's question, the tail elongation, is that has a time scale associated with it? Is that also tied to the same clock as we're discussing here?
No. So this clock doesn't control tail elongation speed. It has its own timer.
Okay.
Excellent question. It's more like a sand timer though, rather than periodic timer. Yeah. Wonderful. So far enjoying it. These are all cool quantitative questions. So, okay. So if our model is correct, and this hierarchical relationship is important, and we can simulate it, and in silico it seems to be good. It's capable of discretizing information. Then we should be able to take genetic mutants where 4 gene copies of clock are mutated or deleted, right? There's no molecular genetic biological clock, no oscillations. All segment borders should be disrupted in that background if we grow those embryos. But if we mimic what clock does to ERK kinase, ERK is a kinase druggable pharmaceutically. Pulsatile inhibit ERK kinase activity. We should be able to bypass the molecular clock. Okay, that's the experiment basically we did. But before we had to do some simulations to encourage our lab members because no one was willing to do it initially. So this is now a clock mutant where ERK activity is static, time is vertical, PSM is horizontal, it's growing leftwards posteriorly. Okay, the gradient is regressing from right to left, and the levels are peaked, the levels are fixed. This is how we see in data as well of the ERK activity gradient. So this is a simulation for in a wild-type embryo where pulsatile inhibited by the clock. Again, the gradient regresses from right to left, anterior to posteriorly, as time proceeds vertically. But now the levels oscillate, right? So our goal was to convert this system to that system while pulsatile inhibiting the ERK kinase. Is this possible? Because we're going to be bathing embryos globally, right? No spatial control. So initially we thought, would it work? That's why we resorted to in silico simulations. Again, if you simulate it, so that the model gives encouragement, doesn't give any proof, right? If you say, let's say 8 minutes perturbation, 32 minutes washout, 10 minutes blocking ERK activity, 30 minutes washout, recovery, various time configurations and various drug concentrations, right? The simulation suggests that there could be some windows in this phase diagram, some areas where the picture might be convertible from this bottom black box to top red box. If this is the real business clock does, and clocks in other hundreds of targets are irrelevant, and the only role of the clock to drive ERK activity oscillation, then we could be able to restore it. Obviously, it doesn't tell us which exact concentration because we don't have a map from a concentration to biochemical rate. So we are experimentally have to tinker to find where we are in that island space. Again, the time period also we had a little bit experimentally tinker. So after several months of experimental working of several lab members, we find the success at that. Okay. So we now took our chromosomal deletion mutants where all segment borders are fractured and fragmented, not intact and proper. We bathed them with the receptor kinase inhibitor of this MAP kinase cascade. We used two different inhibitors inhibiting two different kinases on two separate experiments. In this particular case, 10 minutes inhibition, 30 minutes washout. Okay. So these are chromosomal deletion mutants. Both HER1 and HER7 4 copies are gone in two chromosomes. And we are bathing them with this control solvent, DMSO. It doesn't do anything. All segment borders are still fragmented and disrupted. Siblings of these mutants coming from the same mating clutch. We now passed them 12 times with the receptor inhibitor. We were able to re-engineer these segment borders. Not before our pulses, not after our pulses. If you image them in live, those borders form sequentially. They're not like coming all at once. And the borders that are forming, where are they induced? They're induced in the correct location, that black star, red dot in the animation I was showing you in the middle of the tissue. We know that location based on the distance from the tail tip as compared to control wild-type siblings at the same stage. Okay. So at the correct location, in a pulsatile manner, sequentially the boundaries are instructed. Again, we can do several stainings to characterize these segmented somites. This is the staining for another kinase that is localizing these myopendenous junctions forming the somite borders and control embryos nicely. And in the mutants, the segments are disrupted. The kinase's localization is obviously disrupted, the blue color. In the siblings of these mutants, where we had 5 pulses we gave them, in the same embryo, purple box, before our pulses, the fissures are not there, segmentation disrupted, kinase is mislocalized. After our pulses, yellow box, we can induce these fissures between cells, and we can properly localize the kinase. If you measure the myofiber lengths that are attaching to consecutive segment borders, in this particular concentration and temporal window, we basically reverted them back to wild-type lengths. They're exactly like wild-type. Then we asked, can we play with the timing and change segment numbers and sizes, right? Your question. And yes, we can do that. So we now took chromosomal deletion mutants, split them into 2 groups. The first group, blue population, we give them 8 pulses, 8 frequent pulses, 6 minutes inhibition, 24 minutes washout. The second population, green, we gave them 5 less frequent pulses, 10 minutes inhibition, 40 minutes washout. If you do the math totally calculated, the total duration is approximately close to each other, which means we are targeting the same window of developmental development, embryonic development, and the same body portion of the axis, right? With that, we were able to divide with the first group 8 smaller somites, the second group 5 larger somites, and the layer lengths are plotted on the y-axis on the graph. In separate parallel experiments, we also ask whether can we restore ERK activity oscillations as we predicted from our simulations. So we did immunostaining for ERK activity. The middle panel is for the rapid pulses. The right panel is for the less frequent pulses. ERK activity has a gradient, I told you. It's peaking here in the posterior to mid PSM and declining anteriorly in both cases. So before pulses, the gradient peaks as shown here with orange color. Immediately at the end of our inhibition, 6 or 10 minutes, if we fix embryos, the peak levels, as you see, decreases to purple in both cases. Everywhere, even the slope of the gradient decreases. At the end of the first washout, before we apply the second pulse, the gradient recovers back to black. Okay? Because this pulsatile treatment does not inhibit tail elongation, the tail still elongates posteriorly leftwards. It drags the gradient orange, purple, black leftwards. But as the gradient moves leftwards, the peak levels come down and up in different locations. Okay, sounds good. And the same immunoquantifications, if you do it in wild-type embryos under the control of normal endogenous clock, intact clock, we got the same oscillatory behavior, exactly similar to this. If you do these quantifications, you can also look at the paper. In the clock mutants, now you see the gradients moving posteriorly due to tail elongation, but the peak levels are fixed. They're not oscillating, coming down. Sounds good? All right, so this is the summary of the second portion of my talk. So how spatiotemporal information of somite segmentation is accomplished. We had this long-standing half-a-century old textbook model, which stated that there's a static gradient providing positive information at an unknown concentration threshold. It's acting as a permissive cue, waiting a second cue to come the clock. They're acting independently, and how they integrated, nobody knows, okay? So what we find is that the gradient is not static oscillatory. The clock actually acts first, and then gradient acts next, and it's instructive information, not permissive. And there is a hierarchy between them. Clock drives gradient's oscillatory dynamics. And by mimicking this hierarchical regulation, we can bypass a molecular genetic clock. In the absence of it, we can re-engineer missing segment borders, just rewiring the system in a way that we mimic clock's action on the gradient. With that, we propose this new model. We think this model, although we've done the experiments only in zebrafish, would be valid to other vertebrates too, because at least the Hess oscillations are concerned in mice. ERK oscillation is concerned, and its dependence on Hess genes also concerned in mice as well. They just have not done the same experiments as we've done in the fish so far. But in addition to vertebrates, there are several other invertebrates. They also segment their body axis. They don't have vertebral column, they segment other tissues along their body axis. These are sequentially elongating, growing insects that are segmenting their body axis. They are much less studied than vertebrates, but of the few labs and few species they studied, they also find some molecules oscillate in those species, and some molecules establish a gradient. But their molecular gene identity is completely different than the vertebrates. Now the question is, can we think about a theme or engineering design principle to generate sequential segmentation of body axis? Up until now, we didn't know how it's done in vertebrates even, so that's why we didn't have any idea to propose. But now we basically speculate that perhaps the cog model could be that engineering or design's principle. If their clock genes in the invertebrates also inhibit their gradients, they could also in principle generate sequential segmentation of body axis. Obviously, at this point, this is just a sheer speculation and needs to be tested and scrutinized in invertebrate species by colleagues that are studying in those model systems. So I have presented to you a large background as well as two published stories from my group. The last story I presented to you was led by a former postdoctoral fellow, Mohammed Shimshek, who launched his own lab at McMaster University in physics and molecular biology department early this year, 2024. And he received help from two graduate students, Angad and Dida, who generated the ERK reporter as well as did several immunostaining experiments. Another undergraduate student was involved in the earlier portion steps of the coding and script writing. The earlier project in my talk I presented about gene pairing was led by a former graduate student, Oriana Zinani, who generated the CRISPR-Cas9-based genome engineering. And she received help from a senior postdoc fellow in the lab who did the live imaging for that story. And hopefully Kemal will also be going into job market next year for faculty positions. In the lab, we have positions open for postdoctoral fellows, graduate students. So if you know anyone interested in studying pattern formation, developmental pattern formation, utilizing inter and multidisciplinary techniques, please refer them to me. I will pay your agency fees handsomely. Thank you very much for your patience. I can take questions.
I was just curious, this corn snake you mentioned had like 315 vertebrates. How is that so precisely controlled? Nothing that you just, I couldn't recognize anything.
Yeah,
that
but I mean,
controlled that very well.
in biology, we never have 0% standard deviation, right? We don't have a dot. So there's about 3-4% variability in all vertebrate studies so far, including corn snakes. So we use that in my portion of my postdoctoral studies as well. In that lab, we had corn snakes. In one study, we had used it. The real cause of it, the answer is not known. Whether you have 303 or 30, obviously with higher numbers, you might think error rates might be higher, but your standard deviation can also grow slightly, right? The exact number. The mechanism we do not know. We also so far do not know the precise mechanism defining the number of somites in each species as well. We have phenomenological models, some ideas, but nothing concretely tested so far. I personally think it's the substrate limited system. The embryo generates cells that are allocated to this tissue before somitogenesis during gastrulation stages. And there is a fixed flux coming into this tissue in each species differently. Once they deplete those cells, that's the stand timer I was telling you, the X elongation stops. That's my personal view, but nothing so far has proven regarding the exact precise mechanism controlling the somite numbers and that huge variation across species, and that's precision in each species.
I'm wondering if you have any hypotheses about what might be a functional advantage of this cog mechanism over the textbook mechanism. Because at least as I understand it, it seems like previously you had an oscillating thing. You still have an oscillating thing, but now it's just it's the double-phosphorylated ERK. And previously you had a gradient that you had a fixed gradient, but now it's a fixed ratio. So it seems like
there's a few, maybe. We obviously experimentally testing it is impossible because we have to engineer the textbook version of segmentation, which doesn't have, they can't segment, right? Unless you engineer it or try to engineer a new way of segmenting, experimental testing will be difficult. That's why we'll just give you some speculative answers right now. One is, one difficulty would be, so there is a moving waveform in a static gradient, right? Continuously regressing gradient. And there is a stripe of clock wave coming. So they do not intersect only in one place. They intersect in multiple rows of cells, right? The clock wasn't a single row either. So no one had addressed that deficiency in the textbook model because nobody bothered simulating it, right, in that manner. So that was an issue there. Discretization problem wasn't 100% solved by claiming they provide independent information. Still, multiple rows of cells will be passed through each other at the same time, and they will compete with each other to make the segment. That's one deficiency of the textbook model. Immediately, I can share. Another one is a more maybe difficult rather than deficiency. So now you only need these downstream molecules to read out one pathway rather than two, right? Reading out two inputs that are intrinsically different, one special gradient, one oscillatory input, and precisely reaching out a decision could have been difficult for a downstream molecule, more difficult. As compared to there's a hierarchy, the downstream molecule doesn't need to know about clock. It only needs to know about the ERK activity. So these are the two things I can think of, maybe an advantage or theoretical speculative advantage. Testing it experimentally is currently not in the realms of feasibility. Yes, please.
So I got caught in one sentence that you said there about this is a model, but it's not quantifiable or possible to simulate. And this remind me of that famous article about how a biologist fixes a radio that you may know about, and if other people don't know about it, it's a really fun read. But the idea about what is an appropriate model, right? And there is this tendency, I think, by biologists that don't have a quantitative training to build these models that lack timescales, concentrations, or specific interactions. And you just kind of make a word thing about what could happen, but it's actually not implementable.
Yeah.
And the need to move to things in which there are timescales, there are actually fluctuations and real concentrations and stuff like that. And so how do you think the education of biologists in terms of this and kind of moving away from these non-models to actual models? What would be that we would need to do in training new biologists to make them aware of this?
Before trying to or attempting to address your question, one disclaimer I will repeat, as I said in my talk, but might have been lost. Again, I am grateful to that model, because that model was half a century ago, before discovery of any oscillating genes, gradients, etc. So those guys were really mind-blowing genius scientists, you know. But at the time of the construction of this phenomenological model, I think the problem wasn't the people who proposed it, who was very smart guys, in the absence of any evidence proposed such a thing. The problem was maybe of us, of the later coming ones, as data accumulates, more molecular knowledge is gained, some things are not fitting. Trying to renormalize it, putting under the rug of the dirt and covering it, that was the mistake, I would think. It should have been more concrete attempts to update it. It should have been done earlier. So it's not the fault of the original people who proposed that. In fact, one of them was as a theoretical person. It was proposed by a biologist and a theoretical person about half a century.
The theoretical person didn't try to do computations on it?
He did some things, but again, it was quite an abstract catastrophe-based model, some bifurcation curves he plotted, but nothing non-molecularly to really expand on that aspect. So, but again, now we have more things are known, we sequence every gene, we image many things. So from now, obviously, we should be building more and more testable models and update our models. Now, no models will live forever. So every model will require likely this one too, although we so far happy with it, not see no deficiency, but every model has a limitation. Right? One way of doing it, probably interdisciplinary training and courses, such as in this department or other maybe engineering departments also do that. Building courses, inviting biology students, asking them to take as a prerequisite some of those modeling or interdisciplinary courses. Unless that is done, then the only way to move forward will be forced collaborations.
What about textbooks? Sorry if I'm insisting on this. Because if you take a traditional textbook that is teaching biology to undergraduates, it goes through a lot of descriptive stuff and a lot of the non-model models, right? This does that, but again, there are no time scales, there is no, and how could we integrate the concept? I mean,
gradually. It won't be immediate, like a diffusive process. As biologists see the success of interdisciplinary research, I see now many departments are recruiting quantitatively trained people into biology departments, like my postdoc, right? So this will increase. As they increase, they will set up new interdisciplinary classes within biology departments. Not every class will be like that, and they do not need to be like that. As long as they have some classes, that's adequate. Again, biology is quite vast as compared to, let's say, physics. So you cover a vast area molecularly. Encyclopedia of materials they need to deliver. Obviously, there are other courses should deliver such materials.
Need to deliver.
And then maybe they will also set up some classes, a few, to give quantitative training so that as we gain in other systems such knowledge, they can also be implementing such modeling process. Radical changes is different.
Okay, well, let's thank Ertugrul again.